Recording started.
Hi Bio30 students, this will be our last whiteboard session of the year. We're going to go over some stuff from the, actually I was going to say from the final exam, but from different tests across the year, some different questions that students found challenging. And also I'm just going to kind of go over some other concepts that fit in with some of the concepts we'll go over here. So Mary, if there's anything you want to stop me on at any point and go over, just let me know. And otherwise we'll go from there. So this first question here is, it says in humans when iodine levels are adequate, abnormally high TSH secretion would likely result in. Okay, so let's go over that whole system that deals with TSH. So here's the diagram here, and I think it'd be really helpful for you guys to go over those 3 study guides that I gave out in the beginning. Study guides 1, 2, and 3, they were on the endocrine system, nervous system, and I forget the other one, I think hearing and that sort of thing. So that will give you a really good handle on this kind of stuff that relates to hormones and glands.
You have an email.
Those types of questions seem to be in fairly large numbers on the diploma. Okay, so here's the basic system. So first of all, we know that the hypothalamus is the master gland and it controls the pituitary gland. The pituitary gland produces TSH. And just remember we got the 3 T's here. TSH controls the thyroid and it causes the thyroid to produce thyroxine. And then what thyroxine does is it allows us to produce energy. Basically it's breaking down sugars or glucose and we're getting energy from that. So basically the higher the TSH, the higher the thyroxine, and the higher the energy levels. But remember what's going to be in low amount is actually going to be sugars, because sugars are being broken down. So blood sugar levels are going to actually go lower. Okay? So because greater amounts of TSH, like this question's talking about, are going to produce more energy, the answer for this would be nervousness, so we've got like lots of nervous energy, and we're also going to have weight loss because we don't have a lot of that glucose sitting around and it's going to be stored, it's being used up for energy. Okay, so if we've got high levels of TSH, and remember that's called hyperthyroidism, and hypothyroidism is a small amount of TSH being produced. So you're going to have the opposite. So in that situation you'd have something like sleepiness and weight gain. Okay, so if you want to remember the difference between the two, think of a hypodermic needle as being very small. So hypo is small amounts of something, hyper is large amounts of something. Okay, so just remember this basic system then, that TSH produces in the end high amounts of thyroxine, which produces a lot of energy but low blood sugar because the sugars are being used up. Okay? So we'll go on to the next one then, and again, please stop me at any point. The next one says the response to increased glucose levels in the blood is the conversion of glucose to glycogen. This response is the result of the secretion of what? Okay, so whenever we're talking about either high glucose levels or low glucose levels, we're either talking about insulin or glucagon. They always relate to the glucose levels. Okay, so why would we change glucose to glycogen? There must be too much glucose in the blood, so we need to store it. Now insulin is always produced when we have lots of glucose in the blood. Remember, insulin is kind of like a key that allows the glucose to enter the cell. Okay, so it goes from the blood into the cell and allows the cell to be able to use glucose. Whereas glucagon is when we want to produce more glucose. Okay, so they have, again, opposite purposes. So if we have a lot of glucose, we're going to use insulin to let it into the cell. And if we need more glucose to be produced, we're going to produce glucagon. So because this situation is talking about increased glucose levels, we want to let that into the cell, and so we're going to produce insulin. So the answer is C for this one. Okay? And so specifically, glucose increases the permeability of cells to glucose. So if something is more permeable, it's more easy for it to get into it. All right, so we'll go on to the next screen. This one says the movement of hair cells in normal ears opens tiny pores called ion channels in the nerve cell membrane. This process begins nerve impulse transmission along the auditory nerve. Nerve impulse transmission continues along the nerve cell membrane as, okay, and the answer for this one is going to be a wave of depolarization. Basically, whenever messages are sent from one cell to another, the cells become depolarized. So normally cells are polarized, and then when a message is being carried by the cell, that's a depolarized cell. So because this is talking about nerve impulse transmission, so the message is being carried from one cell to another, it's a wave of depolarization. These diagrams here are just showing us that when you have a resting cell, it has a negative charge on its inner membrane. So again, that's a polarized membrane, but when you have a membrane that's carrying a message, that's called a depolarized membrane, and it's got a positive charge on its inner membrane. And again, the charges on the inside of the membrane are a result of the flow of sodium and potassium. They both have a +1 charge in and out of the cell. It's called the sodium-potassium pump. Whenever we've got a cell that's carrying a message, it's always a result of sodium flowing into the cell. And that's what produces the positive charge on the inner membrane. Okay, so this one then was a wave of depolarization having to do with message being sent. And so the next question here, we've got some nerve cells here, and it says neurotransmitters are released from what area? Well, it's going to be this point here, Z, or Z, I guess we should say, being Canadians, right? Which is the nerve endings. So the nerve endings are where we produce neurotransmitters that send the message from one cell to another. Okay, and this here again is just a recap of basically the different parts of a neuron. So again, the nerve endings are where the hey Diana, how's it going? Just noticed you popped in. How are things? Oh, right on. Good stuff. All geared up for summer? I was asking Mary what she's going to do for the summer. She might possibly go to Ireland. How about you? You going anywhere special? Okay. Well, hopefully it's a good relaxing time for you. As for me, my parents are coming, last day of school, they're coming from Ontario, so that should be a nice time. And then we're going to head out to Oliver, BC. And I've heard they've actually had some mudslides there recently, so I'm hoping that that's still not going on when we get there. But I always love going to BC. I don't know if you guys have been there a lot, but I always just am floored by the beauty that you see there with the tree-covered mountains and just all the rivers and stuff like that. Just awesome. Well, hey, so I'll just pick up what we were talking about with this question here. We were talking about where the neurotransmitters are going to be released in this diagram here, and we said that it's going to be this point here, point Z, because that's the nerve ending. And so the nerve ending is going to send it to the next cell, and this here is just kind of a recap of the different parts of a nerve cell. You have new mail. Dendrites receive messages from another cell, and the nerve endings send to another cell. Okay, and then we've also got the axon. The myelin sheath is kind of like an insulator, insulates the message. So, okay, we'll go on then to the next one. Next one says, using the following information to answer the next question. Okay, serotonin is a neurotransmitter found in the brain. Some studies show that too little serotonin can cause depression and lead to a tendency to behave impulsively. By the way, just out of curiosity, or just for information here, when you eat sugars, it produces more serotonin. So even though sugars aren't always that good for you, they do cause serotonin to be produced, which is kind of cool. So, yeah. Anyways, it says excitatory neurotransmitters like serotonin. Okay, so it's going to be an excitatory neurotransmitter. Well, what does that mean? Well, basically it means it causes more nerve transmission, right? That's what a neurotransmitter does, that sort of chemical. And so we're causing a message to be sent. Well, what type of condition takes place in a cell when a message is sent? Well, we were just talking about it in an earlier question, that whenever a message is being sent in a cell, that cell carrying the message is called a depolarized cell. A resting cell is called a polarized cell. And what causes it to be depolarized is when sodium enters the cell. So what it must do is it must increase the permeability of the membrane to sodium. So anytime the cell is carrying a message, sodium is entering the cell. Okay, so that's going to be the answer. If it was, if we're talking about a neurotransmitter that was like a nervous system depressor, then it would be something like this: D, decreasing the permeability of the membrane to sodium. Okay? All right, so we'll go on to the next one. This one here says, which structure acts as a lever to mechanically amplify sound vibrations? Okay, the answer for this one is going to be structure 2, because this is pointing to some of the bones of the middle ear, and their job is to amplify sound.
You have new mail.
But I just thought that, with some of these questions, I'll kind of tie in some other information just so you have a kind of a rounded view on it. So also, the structure 4 is going to be the tympanic membrane, or the eardrum, right? And we also have structure 1 is going to be the semicircular canal, and that's used for balance. Okay, so that's structure 1. That's right here. That's used for balance. And this structure here is going to be the cochlea, and that's used for hearing. Okay, so that's just kind of an aside. The main part was the bones in the middle ear being related to amplifying sound. Alrighty, so going on to the next part, it says the area of the eye where sensory reception of light is most acute and an area where there's no sensory reception are labeled respectively. Okay, well this section right here is going to be the blind spot, number 4, and the blind spot is right where the nerves go out of the eye, and number 3 is going to be the fovea centralis, and that's the area where we have sharpest vision. So they're actually fairly close to one another, but just remember the blind spot is the part that's right near the nerves. And remember that the reason why the fovea centralis has the sharpest vision, gives us the sharpest vision, is because we have lots of cones there. Remember, cones and rods are both photoreceptors, but cones are responsible for sharpness of vision and also color vision, whereas rods enable us to see in dim light, and they kind of enable us to distinguish between light and shadows, sort of thing. Okay? So, we'll go on to the next part. Next part says, airborne molecules are thought to bind to odor receptor proteins. These odor receptor proteins initiate the smelling process by. Okay, so this one is kind of similar to what we were talking about before. The answer is going to be making the membrane of the neuron more permeable to sodium ions. Okay, so because we've got some sort of message being sent, we know that the cell has to be more permeable to sodium ions. Okay, so we've already gone through that kind of stuff. Next question, I know I'm going over these fairly quickly, and again, please stop me if you need to.
You have new mail.
Next one says, when a patient complained that things far away were out of focus, a possible diagnosis is. Okay, so we've got some different names here. Well, here's what we know. If you can't see things far away, you can only see things near. That means that you're nearsighted. Another name for nearsightedness is myopia. Okay? So the answer for this one would be myopia. The other condition is hyperopia, where you can only see things far away. You can't see things very well that are near. And if you want to try to remember, okay, well, how am I going to remember which is myopia, which is hyperopia? If you remember that M and N are close to one another in the alphabet, you'll remember nearsightedness. Okay, that's myopia. Okay, and then hyperopia is farsightedness. All right, so on to the next one. By the way, are you guys watching World Cup soccer at all? Oh yeah. Do you guys like hockey? Do you like watching sports on TV in general, or because I find definitely I talk to a lot of people who find soccer boring on TV. because there's just not too many goals scored. Personally, I find Brazil, if you guys ever watch Brazil, they're pretty exciting to watch because they get a lot of long ball crosses and stuff like that. But did you watch the NHL playoffs at all? Oh yeah, yeah. I didn't watch too much either. My team was Montreal because I grew up in Montreal, and once they were out, I wasn't quite as interested. But it's personally, I find hockey a lot more exciting to watch, even though I love playing soccer. But if you do ever turn on the World Cup of Soccer, you'll notice that they've got these loud noise every time you watch the game, because the South Africans use these kind of trumpet-like things called vuvuzelas or something like that, and they just sound like a bunch of bees. So if you watch a game, it just sounds like buzzing bees the whole time. It's kind of funny. Anyways, we'll go on to the next one. Next question says, the row that identifies the structures in the male that have similar functions to the oviducts and ovaries in the female. Okay, so we'll just review with the oviducts and ovaries. Okay, so the oviducts are tubes that carry the eggs from the oviducts, or from the ovaries, sorry. Okay, so they're tubes that carry the eggs. And what's similar in males is the vas deferens, okay, because that's a tube that carries the sperm away from the epididymis where they're stored. Okay, so remember that the oviducts in females are similar to the vas deferens in males, and then the testes in males are going to be similar to the ovaries in females because the ovaries produce eggs and the testes produce sperm. Okay, so the answer for this one would be A, vas deferens and testes. And it's also important to be able to label different reproductive structures on a diagram. So just wanted to make note of that as well. All right. So the next question here, it says, which hormone is at its lowest level at the time of ovulation? Okay, so is it LH, FSH, estrogen, or progesterone? So I'm just going to recap a little bit of stuff about different hormones that are involved in pregnancy. Okay, so first of all, so puberty is started by the secretion of hormones by the hypothalamus. Okay, now getting into the hormones then, luteinizing hormone assists in the development of the follicle, which produces an egg. LH is high just before ovulation. Now, LH in a male stimulates the production of testosterone. As far as FSH, remember it's the maturation of an egg, and if you think of the word F for finishing, that's kind of similar to maturation, so that helps us remember what FSH does as far as the egg. Now both estrogen and progesterone are produced all during pregnancy. They're the major hormones involved during pregnancy. Both levels continue to increase until just before birth. Estrogen levels control progesterone levels. Progesterone is the lowest at the time of ovulation. It's the opposite of LH. Levels drop near the end of pregnancy. This is part of what stimulates childbirth. Okay, and here's two other purposes of progesterone as far as the endometrium and the uterus. HCG, okay, that's human chorionic gonadotropin. It's a bit of a mouthful. That's produced until 8 weeks, and the purpose of this is to maintain the corpus luteum. All right, so, and if you'll remember from that, which one is low at the time of ovulation? Okay, that's progesterone. So that would be the answer here. It's going to be answer D. Okay, so going on to the next question, a thick and vascularized endometrium.
You have new mail.
Okay, so what is the endometrium again? Well, that's the lining of the uterus. And, okay, so the answer for this one, a thick endometrium is present when an egg is fertilized and progesterone levels remain high. Okay, so the fertilized egg is implanted into the endometrium, which is the lining of the uterus. Okay? So, going on, this here is asking us to be able to identify one of the structures having to do with the developing fetus. And so it's asking us for, well, it says during labor, smooth muscle contractions occur in structure, and it's going to be structure 2. That's the uterus. And that's what this here is pointing to. So the uterus, as we know, is a thick muscular wall, and contractions in the uterus are going to happen during labor. All right. So, moving along. Okay, this next question deals with haploid and diploid and mitosis and meiosis. I know this might be a little tricky to read here. It says, during human fertilization, blank gametes. Okay, so what kind of gametes would it be? Would it likely be haploid gametes or diploid gametes? Okay, so they're going to be produced by which process? So which process produces? Okay, well, during fertilization, haploid gametes, okay, so those are the gametes that have half the amount of DNA, right? Because gametes are always haploid, right? They are like what we call 1N, and 2N, like that has the full amount of chromosomes, which is 46 chromosomes, that's going to be diploid. Okay, so during fertilization, so I'm just filling in the blanks here, haploid gametes produced by, well, that's going to be meiosis, right? The process of meiosis produces gametes. That's the purpose of meiosis. Meiosis is a type of sexual reproduction. Okay, so haploid gametes produced by meiosis unite to form a diploid zygote. Okay, so the first structure when the egg and the sperm meet, that produces a zygote, which is diploid because it's 2N rather than 1N. And then that zygote is going to have cell division take place over and over and over again until it produces an embryo. And when that cell division is taking place, that's mitosis. Because the purpose of mitosis is either cell division, it's either growth or cell repair. Okay? And so mitosis, unlike meiosis, is asexual reproduction. Alright? So that's what we would put in these blanks here. These here would be the answers that would go in these blanks. Okay? So is everything making sense so far, guys? Okay, cool. So, next question. I know the next one says 15 and it says 13. I wasn't able to change it, or I was just kind of going through this stuff quickly. So if you just want to focus on this as being number 15. Okay, and this one says, so it shows us that the chromosome number of a horse is 66 chromosomes, or 2N. And it says, starting from a single cell, spermatogenesis in horses produces, okay, well the answer is going to be 4 cells. Okay, so whenever you have gametes produced, you have 4 cells being produced. Each of the cells, as we know, whenever gametes are produced, because spermatogenesis is the production of sperm, whenever gametes are produced, they're going to have half the number of chromosomes, as the parent cell. So instead of 66, they're going to have 33. All right? So that's why we're going to have 4 cells and 33 chromosomes. All right. On to the next question. And some of these will probably be just a little bit easier to read on the PDF file than on the whiteboard, because something happens when I put it on the whiteboard. It's just not quite as clear for certain questions. Some of them are fine, like if it's typing, it's okay. Okay, so this one says, a human, excuse me, sorry. A human with Klinefelter syndrome has 47 chromosomes and has the XXY chromosome instead of XY. From what does this abnormality result? Okay, whenever you have an extra chromosome or one less chromosome, it's always a result of non-disjunction. Non-disjunction is a failure of the chromosomes to separate during cell division. Okay, so non-disjunction is what causes cells to have either one extra or one less chromosome. So that's why we have XXY instead of XY. Okay, so the answer would be non-disjunction occurs in either the formation of the sperm or egg. All right. So on to the next one. The next question says, the cell processes diagrammed are more advantageous than budding. Okay, so first of all, we need to figure out what's being diagrammed, and then we need to figure out, what budding is. Well, we see that cells are joining together here, so we assume that this is sexual reproduction, at least at this point here. And then it's saying that these cell processes are more advantageous than budding. Well, budding we know is a type of asexual reproduction. It's used by yeast cells to reproduce. So overall, we're looking at the idea of why is it that sexual reproduction is more advantageous than asexual reproduction. Well, the reason is, there's a couple different reasons, but one is that sexual reproduction involves the recombination of genes from two parents, which produces variety in offsprings. And variety means there's a better chance of survival, right? If you're looking at asexual reproduction, everybody looks the same, so if conditions change, then everybody can be wiped out, right? Whereas with sexual reproduction, you've already got someone who might be able to adjust to the new conditions. Okay? So the answer is going to be A, new genetic combinations are produced. Alrighty. So, next is a diagram of an ovary, and it says, which description identifies the chromosome content of cell 4? Okay, so this one's cell 4. So we've got an egg cell here. So we know an egg cell is a gamete. We know it's got half the DNA, or half the number of chromosomes as a regular body cell. So rather than having 46, it's going to have 23. So we're going to say it's N equals 23. If we talk about a regular cell, it's like 2N equals 46. Okay, so both egg and sperm have 23 chromosomes, regular cells have 46. So whether you're talking about a zygote, that would have 46, or any, just a regular muscle cell, whatever, it's going to have 46. Okay, and let's see if I can read this here. Hopefully I can. It says, many adult newts and salamanders have a remarkable ability to regenerate amputated limbs. Did you guys know that before? It's kind of cool, eh? After amputation of a foot, a newt will regenerate the lost foot. However, if a newt has its foot amputated and receives a particular dosage of vitamin A, the animal grows back a whole new forelimb, not just the foot. It says a logical interpretation can be drawn is that vitamin A must play a major role in. Okay, well, we know that mitosis has to do with cell repair, and that's what's going on here, right? So somehow this must be increasing the amount of mitosis, and so it's got to be answer A. All right? And the next question says, which expression of chromosome content represents somatic cells in people with trisomy disorders known as trisomy 21? Okay, so trisomy 21, or Down syndrome, is when you have an extra chromosome. So rather than being 2N, it's 2N plus 1. So it would be answer D. All right. Well, Mary, I'm like you. I've got a sore throat here. I think I've got a bit of a cold as well. You know, one thing I find, I don't know if you guys have ever tried this before when you get a cold, but I find if you gargle with salt water, and I don't know whether this has been medically proved or not, but personally I find it works. Gargle with salt water, or if you actually take a syringe and put some up your nose. I know it sounds kind of gross, but I find it seems to help with a sore throat. So, and I don't know if do you guys ever try Cold FX? I'm not sure what to think about that stuff, but I know a lot of people like that stuff too. Yeah, right on. You find it helps, eh? Oh yeah, yeah, you were mentioning that before. That's cool. What kind of essential oils? Like rose hips or something? Tastes nasty, eh? Oh, thieves? Okay. All right. Well, just pop back in when you can, okay, Diana? I think I know what you mean. Cool. Okay. So I'm just going to go over with this next question here. Maybe let us know when you're back, Diana. But we're just going to carry on for now, and if you want to come back to this part later on. So this is talking about different types of cells. We've got polyploids. Okay, we've got diploids. That's 2N. Call it a triploid if it's 3N. Tetraploid is 4N. So this here says if a diploid plant and a tetraploid, in other words, if a 2N and a 4N plant were crossed, the chromosome number in their offspring would be, okay, so the offspring is going to be 3N, so it's halfway between the diploid and the tetraploid. Okay? All right. So we'll go on to the next one. Next one's kind of similar to another one we were looking at before. It shows different animals and different numbers of chromosomes they have in their regular body cells. It says which statement is correct. So I'm just going to go right to the one that's correct here. Again, we know that somatic cells, which is another name for regular body cells, are 2N. And so a sperm cell of a dog, since a regular dog cell has 78 chromosomes, a sperm cell would have 78 divided by 2, which is 39. So answer C would be right. Spermatid, just another word for sperm, would contain 39 chromosomes. Okay? So this one here says, so it's showing some different stages of cell division in the life cycle of a horse. It says, which process illustrates reduction division as in spermatogenesis? Okay, well, we know that spermatogenesis is the production of sperm, and that's, so in other words, that's gametes being produced, so that's going to be process number 4. Gametes are being produced, and again, that's in meiosis. Okay, so it's going to be D, number 4. Okay. Are you with us again, Diana? Okay, awesome. Right on. So next question says, genes which are overruled by other genes which determine the genetic trait are said to be, okay, this is going to be recessive genes, because recessive genes are overruled by dominant genes. This is going to be answer B. And the next one says, traits that are clearly observed as distinct differences are called. Okay, so some kind of traits can be what you call either-or traits, and those are known as, and I just messed this up, those should be discontinuous. Okay, discontinuous, I'm just going to make a correction here. Discontinuous are either-or traits. Okay, so if you're talking about, you know, if there's only a possibility of a creature having a tail or not having a tail, those are discontinuous traits, whereas continuous traits are things like height. You don't say someone has height or they don't have height. It's more like a range, right? Just like with weight. Those are called continuous. So it's like a continuous range, right? Okay? So traits that can be clearly observed as distinct, so it's either one thing or another, those are discontinuous. Okay? All right. Going on to the next one. All right, so this one here, we're looking at some different alleles. Remember, each of those letters are alleles. And we're looking at a situation here where we've got 2 letters being involved. We've actually got 3 different letters, but for this particular question, we only have to deal with 2 of them. Okay, so we've got a curly-haired 5-digited male. Okay, so let's see what that is. Curly hair is going to be 2 little S's. 5-digited digits is going to be 2 little D's. Okay, so the male, you can see here, 2 small S's, 2 small D's. Okay, cross with a female that's heterozygous for both hair type and digits on pause. Okay, again, heterozygous means a mix of alleles, one dominant, one recessive. Okay, so the female is going to be this here. So these 2 are going to be crossed. The question says, what's the probability the mating will produce offspring that have curly hair and are heterozygous for number of digits on pause? So curly hair is going to be 2 small S's. Heterozygous for digits is going to be big D, little d. Okay, so what's the probability of getting this when you have these 2 parents? Well, let's just focus on the S's first of all. So if we focus on the S's, what's the chance of getting 2 small S's? It's going to be 1/2, because half the kids are going to have big S, right? The other half of the kids are going to be 2 small S's, right? Okay, so half the kids are going to be 2 small S's. And as far as the D's, what's the chance of getting big D, little d? Well, half the kids are going to be big D, little d, and half the kids are going to be 2 small D's. So the chance of as far as the S's is going to be 1/2, and the chance as far as the D's is going to be 1/2. And because these are two independent events in math, we say we always multiply the two probabilities together. So when we multiply 1/2 times 1/2, we get 1/4, or 0.25. So that would be the answer for this one. Okay, so does that make sense how we did that? Okay, good stuff. And how are we doing for time now? We're at 11:10, not too bad. All right, so we got a little family tree going on here. It's talking about the incidence of opalescent dentin. Okay, so it's when you've got insufficient enamel on your teeth. And what we need to figure out is what type of genetics. Oh, we lost Diana. Hopefully she comes back. Okay, we got to figure out what type of genetic pattern is going on here. So we need to figure out, you know, is it a dominant pattern? Oh, hi Diana, we lost you there. Got kicked out? Ah, that's too bad. Okay, so we got to figure out what kind of genetic pattern is going on. You know, is it dominant? Is it recessive? Is it X-linked? Well, first of all, we know that most patterns that we see are a dominant pattern, right? X-linked pattern. If we have an X-linked pattern, I don't know if you guys remember this, but if we have an X-linked pattern, we're going to see more males affected than females. And you might remember, if you remember back to hemophilia, the reason is because with hemophilia, you can have like X big H for a normal person, and then X little h for someone with hemophilia. So that's what a female could get. And so if she's got the big H, we say that big H is covering the recessive allele, whereas with a male, all he will get is he will get the X, and if he gets a small h from his mom, that's not going to be covered because the gene he gets from his dad is going to be a Y, and so because of that, more males are going to have the affected gene, whatever it is, if it's an X-linked trait, right? But when we look at this here, we don't see more males getting affected, because affected is shaded, and we're seeing, if anything, more females. Okay, so this is definitely not an X-linked trait. And what you can do for this type of question, we're not going to go through everything, but you can kind of test out to see whether it's a dominant pattern or not. If we have a dominant pattern, then we have something like this, you know, big T, little t being crossed together, and that produces recessive genes. Okay, so we've got dominant parents producing kids with recessive genes. And this is what we see going on here. And so we suspect that it's a dominant pattern, which would be answer C. Okay? So that's what you can do for these types of questions, is just test out and see what the different possibilities are, what they make, if they make sense for each type of pattern. All right, so now we're talking about two individuals within this family tree being crossed. And the ones we're talking about are these two here. Okay, so we've got an affected female and a non-affected male. Okay, so since we're talking about opalescent dentin, let's use the word, letter O to stand for someone who's got the dominant trait, because in the last question we decided that it had the dominant trait, right? There was a dominant pattern going on. So since we've got an affected female, we're assuming that she's going to be big O, little o, because it makes sense that she's got recessive genes going on because they're being passed on, right? That's what we see in her kids. So this is the genes of the mom. The genes of the dad, he's unaffected, so he's going to be two small o's. Okay? So they're being crossed, and the question says, what's the probability the child is going to be a female with opalescent dentin? So, in other words, what's the chance of being an affected female if we do this cross here, using these genes? Well, first of all, we find the chance of being a female, and that's 1/2, right? So we find that probability first. Then the chance of being an affected female. Well, the chance of being an affected female is going to be 1/2, because 1/2 of the time you're going to have big O, little o. Okay, so the chance of being female is 1/2, the chance of getting affected genes is 1/2. So again, we multiply the two together and we're going to get 0.25. All right, so that's how we do this one. So is that cool? Okay, good stuff. Right on. So the next one says, in a genetic cross between two individuals whose genotypes are big B, little b, the genotypic ratio of the offspring will be what? So what we need to do is we need to set up a Punnett square, right? We've got big B, little b. So this might be for the mom, this might be for the dad. And then we would find all the genes, and we would see that we've got one of two big B's, one of two small B's, and two of them are a mix. So that's going to be this answer here. So we would choose this one here. Okay? So that's all we need to do is set up the Punnett square and then see what the situation is for the genes. And remember again, the genotypes are the actual genes. They want you to list the actual genes, whereas phenotypes are, what characteristics they are. Are they, curly haired? Are they, black or white or whatever? Okay. So, going on to the next one. This says, blank is an application of genetic engineering in which genetic information from one organism is spliced into the chromosome of another organism. In other words, it's joined together. Well, this one is recombinant DNA, okay, and I'll show you what it looks like. So here's recombinant DNA. It's kind of a cool technology, because what you can do is you can take some human gene, and if you want to produce that human gene over and over again, like let's say you're trying to fight cancer or something like that, and you find a human gene, well, of course, humans reproduce a lot more slowly than bacteria, right? So if you want to produce a whole bunch of human genes, what you can do is use some sort of enzyme that cuts a chromosome and isolates one particular human gene and then joins that together with some bacterial DNA. So you're also going to cut the bacterial DNA, then you join the two together, and this is called recombinant DNA. And again, it's a way of, like genetic engineering and stuff like that involves this. And it's a really, really interesting technology. So, you can also do it with things like, you could take the gene from a firefly, put it into a plant, and you could get a plant that glows just like a firefly does. So that's pretty cool as well. All right. So the next question here says, a series of enzymes called blank fuse or join free nucleotides in the complementary chain of DNA. So let's just look again at the different enzymes that are involved with the DNA replication. So we've got, first of all, what DNA polymerase does is it's going to join these new fragments of DNA. So when we're replicating or duplicating DNA, we first unzip it, and then we join a new fragment of DNA, and DNA polymerase is what's going to join that to the existing fragment of DNA. Whereas something else called DNA ligase is going to join these fragments together. So just looking at it a little more closely, here's DNA polymerase. You can see you've got your existing DNA. It's going to join these new bases to form a new section of DNA. That's what DNA polymerase does, and DNA ligase is going to join all these fragments together. So for this one, it says it's going to join them in the complementary chain. That would be DNA polymerase. Okay? So the next question is talking about the different bases, right? Remember these things, adenine, cytosine, thymine, guanine, and a lot of times we just recognize them by their letters. Those are nitrogenous bases. And so just to review, DNA is made up of lots of nucleotides put together. Well, what's a nucleotide? Again, a nucleotide is a phosphate, a sugar, and a base connected together. And the bases are the part of it that are kind of the center of the DNA. Now, if you're talking about just part of a nucleotide, if it's just the phosphate and the sugar part, that's called a nucleoside. So it's only when you add in the base part that you have a full nucleotide. And again, a nucleotide is like the building block of DNA. And it's made up of phosphate, sugar, and base. Okay? but this was actually asking us what adenine, cytosine, etc. are, and those are the nitrogenous bases. Okay? And let's see how we're doing for time. We've got about 10 minutes. Good stuff. So I don't think we're going to get them all figured out. We won't get them all finished today, I should say. So I think what we'll have to do is I'll just make up a separate one for the ones that we don't cover. Okay, this one here is talking about crossover frequencies. And you might remember that we have to make up maps when we've got these different frequencies. It says the crossover frequency between gene E and R. So when we look at the information here, we see that gene E and R, well, here we can see that gene F and R have this percent crossover frequency, and we can see that F and E have this percent crossover frequency. Well, remember, the further apart two genes are, the higher the crossover frequency is. So what this means is that F and R must be further apart, and the F and E must be somewhere in between F and R. So what we can do then, to find the difference between E and R, we said that F and R have a difference of 4.5, we call map units or percent. F and E was 2.5%, and we want to find the difference between E and R. So we're calling this question mark, and we can see that it's going to be 4.5 minus 2.5, it's going to be 2%. So it's going to be answer B, 2%. Okay, so that's kind of how we figure it out. And again, using that, you can come up with a whole map of what the genes look like. And if you want to review how to do that, you can look in the textbook. Sorry, I can't remember exactly where it is, but if you look up frequency, crossover frequency. I just noticed that Mary's typing something there. So I'll just wait a sec. Oh, hi. Is everything okay? Nobody injured? Oh, that's good. Right on. Wouldn't want to hear that your speaker fell in, some water or something like that. You got electrocuted. Oh, that would not be good. Anyways, okay. Use the following information to answer the next question.
You have new mail.
So this one here says we have a small segment of DNA, and we've got a complementary section of RNA. So it says if structure 4, so structure 4 in DNA is adenine, what is structure 8 going to be in RNA? Okay, well let's just kind of review what the base pairing is. We know that in DNA, A and T always pair together. You just remember the word at. But instead of thymine in RNA, we have uracil. And the pairing of G and C is the same. It doesn't matter, excuse me, I'm sorry. It doesn't matter if you're talking about DNA or RNA, you always got G and C pairing together. So, if we're talking about DNA, structure 4 is adenine, then instead of being A pairing with T, if this had been another DNA molecule, because we say that A pairs with U with RNA, this has got to be U, which is uracil. So the answer would be A, uracil. Okay, so does that make sense? Okay, so again, because A always pairs with T if it's DNA, but because this is RNA, A pairs with U. All right, we'll just go on and use the following information to answer the next question. So this question says you've got some DNA that you're starting with, and it asks you which amino acid is going to be produced from this. Okay, it says that you need to use this chart. It's called the RNA codons and their corresponding amino acids chart. Okay, so that's going to be at the back of the diploma exam. So you're going to need to use this chart. And what you're going to need to do is we know that DNA produces RNA, mRNA, and that mRNA is then used to produce the protein. So what we need to do is we first of all need to take this sequence of bases and find out what the bases would be in RNA. Once we find out what the bases are in RNA, then we can use the chart to find out what proteins are going to be produced, or rather, sorry, what amino acids are going to be produced. And then we'll be able to choose one of these. So I'll just show you on the next page. I did that already. I did the pairing here, and so, again, this is U rather than T. And I just kind of underlined each three-letter sequence, because each of those three letters is going to help us find the appropriate amino acid on the chart. So for instance, we've got UUC, and if you look on the chart, you should see UUC is going to be phenylalanine. Okay, that's going to be this here. So that's going to be the first one produced. So already, just doing that, we'd be able to find that it's answer B. We wouldn't have to do anything further, unless there's another possibility for UUC, because sometimes there are. Sometimes you can get more than one sequence being produced. But just in case, we go on to the next one and we say, okay, that RNA sequence would be CCG. Well, CCG is going to be proline, which is right here. This should be, yeah, this here is CCG. Okay, so that's the way you would figure this out. You take the DNA, you find out what the corresponding RNA bases are, and then you use this chart to find what the correct amino acids are going to be. So, we're actually fairly close to the end too. Okay, so this next one has a whole bunch of stuff. I'm not going to read through all this stuff. I want to kind of focus on the gist of it, because it says here, the sequence of events that produces the enzyme, okay, enzymes are proteins, in a liver cell. Okay, so what sequence of events are going to produce a protein? Well, basically we're needing to look at the whole process that's involved, starting with DNA to a protein. And we need to figure out which steps come first. So I'm just going to recap that first of all. So we know that DNA produces RNA, and that's called transcription, right? And then RNA is used to produce proteins, and that's called translation. Another thing to remember is that transcription takes place in the nucleus of the cell, whereas translation takes place in the cytoplasm of the cell, and a specific place is a place called a ribosome. Okay? But these are the basic steps we're needing to focus on. And I'm just going to go into a little bit more detail on the next screen. This is just showing here, again, this is in the nucleus, this is in the cytoplasm. And this is showing more specifically what's happening when proteins are being produced. Again, so you've got the ribosome here, you've got, this is messenger RNA, and these guys are called transfer RNA, and they're bringing those little three base sequences to match up with the three base sequences on messenger RNA. And they're carrying the amino acids. And when those amino acids join together, that produces the protein. Okay, so we know that overall we're going from this to this to this. And so the sequence of steps is going to be, first of all, information from DNA is used to form mRNA. Remember, we're dealing with mRNA before tRNA, and you might just remember, M comes before T in the alphabet. Then we have, so mRNA is going to do something. It's going to carry the message to the ribosome, because that's where translation is going to take place. Okay, so we're going to have 2, then followed by 3. The next thing that's going to happen is transfer RNA is going to transport the amino acids to the ribosome. That's the next step. And you can see all these steps in the diagram up above. And then eventually, the last step, the ribosome encounters a terminator or stop codon. The stop just says, okay, stop producing these, stop, putting together this protein. That's basically what happens at the very end. Okay, so that's why it's going to be 2, 3, 1, 4. Alrighty, so again, I think we're really close because we're on the 40th screen. There's 46 in all, and sometimes the last 2 screens are just kind of advertising. But it is 11:30. I'm trying to think when the next session starts. I think it might be 11:45. So we actually might have a little bit of time still to play with here. So I think we might be okay for now. Alright, so it says use the following information to answer the next question. So we're looking at 2 different processes here, right? We've got the first one, we've got DNA, and it's being used to produce RNA. We know this must be an RNA because it's got the U's involved, right? So this has got to be transcription. Then the next thing is taking RNA to produce all these amino acids being joined together. In other words, this is translation. So process 1 is transcription, process 2 is translation. And again, as this answer here says, answer 1, transcription is in the nucleus, translation is in the cytoplasm. All right? So, this one here, I won't go into it because it's very similar to the other one we had. Just remember, for this we would just take this DNA sequence, we would find a matching RNA sequence, and then we would use this chart to find out which 2 amino acids it's going to be. Okay, so I won't do that just because it's so similar to the last one we did a few questions back. Okay, this question says, when individuals participate in extreme sports, their neurons release more dopamine, which results in a pleasurable sensation because, okay, well what's going on is, so neurons are releasing dopamine, so that's a neurotransmitter, and that's going to somehow cause some sort of a cell to be excited, right? What causes a cell to be excited? That's when depolarization occurs, going from polarized to depolarized, and so that's going to be a neuron containing dopamine receptors is going to reach threshold depolarization. Okay? Now the next question says, in normally functioning cells, the protein coded by a certain gene is produced continually. The process by which the gene's code is read from DNA and the name of the molecule formed by this process. Okay, so we're looking at reading from DNA, so that's transcription, and then the name of the molecule formed in the process, and that's going to be mRNA. Okay, so transcription is when we're reading from DNA to produce RNA. The first RNA we produce is a messenger RNA. So that's going to be answer A. Okay, so I appreciate your patience, guys. I know we've gone through quite a few here. So we've got some sort of life cycle going on, and we've got 2 different processes involved. First one here, we can see we're having gametes produced. Okay, so what process produces gametes? Well, we know that's meiosis, right? So process 1 has to be meiosis. So we're looking at either answer C or D. And so since we don't see gametes being produced here, we suspect this is probably mitosis. And mitosis is cell division over and over again. This is producing a whole bunch of different small cells, and you can see then one of them is being, I guess, differentiated to produce a blood cell. So that's why this is going to be meiosis. Gametes are produced, and this is cell division, so it's mitosis. All right. So, next question says, a possible reason that this parasite may have resistance to chloroquinone is that a certain gene codes for a protein. Okay, I'm just going to underline that. So this gene codes for a protein that plays a role in membrane transport. So if researchers want to develop compounds that are going to block this resistance mechanism, okay, so what you're trying to block is you're trying to block a gene coding for a protein. So how are we going to do that? Well, a gene coding for a protein is basically DNA going to RNA. Well, which of these answers describes that? That would be this here. Because if you can't transcribe DNA or take DNA and produce RNA in the beginning, you're never going to get a protein out of it. So if you can block this from happening, you're going to block the protein from being produced. So this is kind of what best describes what's going on in this part that I underlined here, DNA coding for a protein. Because if you can prevent transcription of the gene, you're going to prevent that protein from being produced. These other answers here don't really relate exactly. Like stimulating translation of the gene, you don't translate a gene because you only translate when you're at the situation of going from RNA to a protein. You don't translate from having a gene, which is DNA. Okay, so that would be the answer to that. And then, let's see, I just want to see how many we... You know what? I think this is the last one. It is. Good stuff. All right. Although I do have a few other questions I might send over on a recording, but there probably won't be too many, so this is most of them. All right, so this is talking about a symptom of MS, and it's talking about some events that take place. So we're talking about motor neurons, sensory neurons, interneurons, and a light receptor stimulating. All right, well, this has got to be the reflex mechanism, you know, that involves these different neurons going on here. You might remember there was a diagram we looked at with motor neurons, sensory neurons, and that kind of thing. So we know that it's the reflex mechanism, and on the next page I'm just going to recap how that takes place. Okay, so what happens first of all is you have a stimulus. So you're at the doctor's office, you get knocked with one of these things on the knee, and so that causes a sensory neuron to fire. That makes sense, that a sensory neuron is going to sense something, right? And then the next one is going to be an interneuron, and then the last one is going to be the motor neuron. Motor neurons control movement, and so that's going to cause your leg to swing forward after getting hit on the knee. So it's going to be stimulus, sensory neuron, interneuron, motor neuron. So when we look at the answer then, it's going to be, first of all, you've got the light receptor stimulated. That's going to be the stimulus. So answer 4 is going to be the first one. Next is going to be answer 2, sensory neuron. And then next is going to be interneuron, 3. And then the last is going to be motor neuron. All right? So that's it, guys. That's a number of the questions that I found people had found challenging in tests from across the year, including the final exam. I'm just going to send over a few later on, but I think these are some important ones to go over. So if you guys can keep practicing these ones, I think it'll really help for the diploma exam. And just a few other last-minute hints for the diploma. You know, one thing is just, you know, always, of course, to make sure you answer all the questions. You know, multiple choice, of course. Guess is better than no guess, right? I'm sure you guys can hear I'm getting lots of mail here. Anyways, another thing, just always remember to go by the guide. You know, if it's a 5-mark question, you might generally want 5 points. You know, that sort of thing. So base your response on the number of marks, of course. And you know, spelling and grammar do count somewhat as well, so check it as well. So those are some of the main things. Also, again, just focusing on the study guide for the hormones and glands sort of thing, because those figure in quite a bit on the diploma exam. So yeah, other than that, I think I will let you guys go. And thanks a lot for checking this out. And so I'll just send over the recording later on, and hope things go awesome on the diploma. And for summer as well. Great having you guys in the class. Hey, you bet. My pleasure. So take care, guys, and have an awesome day.